What is cloning? Cloning might seem like something straight out of science fiction, but it's more real than you think. Most of you have probably heard of Dolly the sheep. In 1996, she became the first mammal to be cloned from an adult cell. While other organisms such as frogs, mice, and cows had all been previously cloned from embryos, cloning from an adult cell was much more difficult. This breakthrough paved the way for many advances in the medical field, including the treatment of hemophilia and mitochondrial disease. While Dolly was the most famous clone made in a lab, cloning has actually existed in the natural world since the dawn of life, from asexual bacteria splitting to identical twins. So what exactly is a clone? A clone has the exact same DNA sequence as its parent, so they are genetically identical copies of each other. So how is a clone created? Scientists use a method called somatic cell nuclear transfer. A somatic cell is any cell in your body that is not used for reproduction. First, the nucleus is removed from the somatic cell of a subject organism as well as from an egg cell. The somatic cell nucleus is then inserted into the egg cell. Now the recipient egg has the complete DNA from the subject organism. Another method involves using electric pulses to fuse the somatic cell into the enucleated egg. Either way, the egg cell with its new nucleus behaves just like a freshly fertilized egg, eventually developing into a clone embryo that is implanted into an adult female. Finally, she gives birth to an animal that has the same genetic makeup as the donor of the original somatic cell, as in the case of Dolly the sheep. What are the uses of cloning? While we aren't able to clone a human yet, animal cloning can allow farmers to breed animals with the desired traits, and scientists may be able to save endangered species. Organs could even be cloned to create transplants, helping those who are disabled or injured. Cloning can also help genetic research, allowing scientists to analyze the composition and effects of certain genes and test drugs on animals with identical DNA. So why is it so controversial? Every new technology has its drawbacks. Cloning is still very expensive and inefficient, so it's unlikely to be used widely in agriculture. And while it can help endangered species, it's detrimental to biodiversity by making organisms genetically similar, weakening the species' ability to adapt through evolution. It also raises numerous ethical issues. Would human clones have the same rights as real people? Would cloned organs create a black market? Is the destruction of stem cells and embryos justified in the name of science? Would we lose our individuality? These are just a few of the questions scientists must consider as we progress towards an exciting future.